This is a production of Cornell University.
Thank you, Gareth. I may be the chair of NBNB, but I love being a member of this field. So thank you for having me. Thank you for sharing your students with me. It's been a great 10 years, and I look forward to stepping down as chair and being a more active member of your field and getting back to enjoy more of my science. The themes of research in my lab are multiple. I'm interested in how volatiles mediate interactions across the full spectrum of positive, neutral, and negative interactions. So we look at how volatiles might mediate obligate mutualism, whether there's private channels that evolve between plants like figs and fig wasps or yuccas and yucca moths. We also study, and this is like obviously an honest form of communication between two intimately tied mutualists. We also study dishonest communication. These are not flowers. These are splachnum dung mosses. And so they are, along with cycads and a few rushes, some of the few examples of plants that have independently evolved flower-like colors of combinations of color and scent and other traits that attract animals to move their propagules, in this case spores instead of pollen. We also study sort of third parties in the evolution of evolutionary shifts of phenotype. Many of you know Kyle Martin, and his thesis work is involved in whether microbes lend a hand in shifts between sort of classic beetle and bee pollinated flowers to flowers that mimic carrion or feces or rotting or fermenting fruit or yeast. And finally, these night blooming flowers that I'm going to talk about today, which are found throughout the world and are pollinated by hawkmoths. Those are the first plants that I studied in my thesis and postdoc, and largely because they're so strongly scented. At the time, the analytical methods I used were not very sensitive, and they gave me the best chance to really confidently sample volatiles. Now we can sample almost anything. So here's the outline for today's talk. I'm going to introduce you to linalool is a ubiquitous, common scent compound that just happens to be a major player in night blooming flowers. So I want to set the stage a little bit for understanding this compound and its distribution. I want to talk about the project. This is the first talk that I'm giving on a project that's just about to end 5 years of funding from NSF on a program called the Dimensions of Biodiversity. It's a big collaborative project, 5 PIs, $2 million, 5 years. It's a chance to really build something. And so you're going to get the first glimpse of what we've been learning from this project. So I'm going to set the stage for you of how we designed it, what kind of questions we were trying to answer. And then I want to home in on one single endemic species, prairie species in Colorado, Enothera harringtonii, which I've brought along with me as I speak. I'm going to pass this around. It's nice to see the plant. Which we work on trees or cycads, you can't always do this. But this is Enothera harringtonii, star of today's show. And I'm going to drill down deep into how Enothera harringtonii interacts with its friends and enemies across its known distribution in Colorado. And then I want to finish up by scaling up to the phylogenetic scale and looking at the genus Enothera, at least in Western North America, and asking if some of the patterns and processes that we think we're discovering in Enothera harringtonii are also shared with its closer and more distant relatives. So that's where we're going today. Let's begin by talking about linalool. How do I know anything about floral volatiles? Where do we start? There are many ways to concentrate and collect volatiles. What I'm showing you here is a kind of equilibration method. For today's talk, we actually collect odors over time, so we get emission rates. But the whole idea is that you trap or adsorb volatiles onto a substance that you can either elute with a solvent, or you can strip thermally into a gas chromatograph. And so what we do is when we collect our volatile samples, which we optimize through trial and error for a given species, we inject them into the GC. I use 2 different kinds of GC columns to separate complex blends. And then we always need a detector, and the detector we use is a single quadrupole mass spectrometer. And so if I do a good job, my chromatograms look like this, and each peak, this is time and temperature. this is abundance of signal. And so each peak, presumably, is a single compound. And how do you know what that compound is? You get the mass spectrum. And the mass spectrum provides you with a hint. In this case, this is a classic mass spectrum for today's compound of interest, linalool, which is a monoterpene alcohol. But getting a mass spectrum isn't an identification, it's just a suggestion. What you do then is you buy the standard, or you make it if you can't buy it, and you co-inject it with your sample. And if they share a retention time, you do it again on another compound, another column, another stationary phase. And if they're always co-eluting, and then you go and look at the possible isomers or the other compounds with similar mass spectra, and you demonstrate to yourself, to your own satisfaction, that they're not all co-eluting at 11 minutes or whatever this is. Okay, and so you can go on to use IR or NMR to identify these things, but there's a kind of Beatles' greatest hits of floral volatiles. There's 1,700 that have been identified, but the same 10 or 20 just show up all the time, and linalool is about number 6. Okay, so although today's context is night blooming flowers pollinated by moths, you can find linalool in all kinds of diurnal flowers, in orchids, in early daphnes blooming in the Mediterranean, etc. So one take-home message is going to be that the roles of floral volatiles are often context dependent. Their ecological functions often have to do with dosage, time of day or night, who's the receiver, what's the community context, what's it associated with, honest or dishonest pollination services, etc. Okay, so if we think we have linalool, then we need to start looking for patterns. I always want to do justice to Roman Kaiser. There are literally thousands of great chemists over the last 50 years who allowed us to use capillary GC&M and mass spec as a way of identifying volatiles. But really only one of them translated the vast knowledge from perfumery and essential oil research into floral biology, and that was Roman Kaiser. This is a guy who worked for Givaudan, a perfume company in Switzerland, traveled the world looking for new odors for his company, and had carte blanche from his employers to publish his work. So he wrote these gorgeous books where he shows the organic synthesis and he shows the chromatography, and he was so far ahead, he was so Mozart to everybody's Salieri's that he just shared everything. And so as a graduate student, I met him and I learned a lot from him that I couldn't have really taught myself. So what Roman Kaiser did first is he hit all of the usual suspects for perfume. People like white night blooming flowers. They smell great. Coffee, citrus, honeysuckle, gardenia, jasmine, etc. Okay, and so he kind of did his own mental principal components analysis and found there's this guild across the planet of white semi-tropical night blooming plants that all smell like gardenias. And what he found is that there's a core of linalool, neuraldol, farnesol. These are terpenoid alcohols. Okay, so they're soluble in nectar, so they can be flavors as well as scents. And then to that core, he found things like aromatic alcohols and esters, lactones, etc. And that adds some of the spice and difference to these different compounds, to these different floral chords. But linalool was almost always there. And when I became a grad student, this is what I read. I did a study of my own with a National Geographic grant and a Fulbright. My first sabbatical was in South Africa about 12 years ago. And we were comparing guilds of night blooming hawkmoth pollinated plants at the same latitude on opposite sides of the planet in Argentina and South Africa. And what we found was that linalool was present in a lot of these plants, and their floral tubes and their morphology was all, sort of a textbook example of convergent evolution. Hawkmoths are universal, they can fly across oceans, and so they're sort of the kind of selective pressure that they bring upon flowering plants doesn't really differ that much between islands or continents, etc. And at that time, I became aware that linalool was chiral, and just because I was finding it in my chromatography didn't mean I knew which linalool it was, and I had to invest in a chiral GC column. And in fact, many compounds that flowers produce as volatiles have chiral centers, and because insects respond differently to them, their receptors are often tuned to the left-handed or right-handed molecule. You kind of have to go that extra step and identify which enantiomer or enantiomers you're working with. So this is just a slide to show you, I'm not going to dwell on the biochemistry very much today, but the structural genes that encode linalool are a part of a huge gene family, the terpene synthase family. The one I worked on as a grad student in Clarkia Brewery with Iran Pechersky was an S+ linalool synthase. And then the ones that are more common in the literature from tomato and mint are an R- linalool synthase. And they are not each other's closest relatives as in this gene family. So this is just to show you that many structural genes and their enzymes that are responsible for the biosynthesis of specific volatiles are evolving repeatedly, okay? And so putting a name on them, linalool synthase, doesn't actually mean anything in terms of their sequence similarity. There's a lot of homoplasia. There's a lot of repeated and parallel evolution in this gene family, okay? There are many ways to make these compounds, in other words. All right, so there's the introduction. Now let's talk about hawkmoths a little and why this matters. You can't read a textbook on evolution without Darwin's star orchid from Madagascar and its 23-centimeter nectar tube and the predicted hawkmoth, Xanthopan morgani. And there have been a lot of people, including Anders Nielsen, who have based their careers on following Darwin and proposing that, well, okay, this is an example of Fisherian runaway selection, but it's not sexual selection, now it's flowers and pollinators. And so how do you get a tube that long and a tongue that long? It might be this cycling of, you withhold the nectar, you make the animal shove its head into the flower more closely so that it transfers pollen more effectively, and that then mutants of the moth that have longer tongues are more effective at doing that, and the plants and moths are stuck in a cycle of longer and longer tongues and tubes. This is in Madagascar. These things happen on islands. This made a lot of sense in 1993. And so we've done a lot of work on the moth side of things. This is my first grad student, Joaquin Goyre. And what Joaquin did in a kind of Tinbergen-esque way was work out what's the whole sequence of it. Once you get a motivated hawk moth, what are the sequence of events that gets it to put out its tongue, approach a flower, and drink? And these are all experiments that Joaquin and I and others have done together over the years and published. And floral scent is involved in many of these early stages of getting a moth to recognize a bright object as a flower, as well as guiding its tongue. And linalool is one of the compounds that we've shown, and others have shown, like Jeff Riffle, to sort of release this searching and probing behavior in moths. So in fact, while I'm talking about linalool, I want to pass around some linalool for you to sniff. Some of you are familiar with it. It's in bergamot oil, Earl Grey tea, Fruit Loops. You can pass these around. And this is the racemic mix for those of you who really want to know which enantiomer it is. Okay. And so we learned a lot over the last decade about how hawk moths respond to flowers and how they might effect that kind of selective pressure on longer nectar tubes and strong overstated floral scents. The problem is, in real ecology, just as in courtship between animals, third parties make a difference. There's a lot of eavesdropping that goes on out there. So guppies, well-known John Endler studies, where guppy males that live in streams that don't have cichlid fish have the most wonderful orange, long, flowery Fantasia tails, and they attract females and win them as mates. But when the cichlids are present as predators, they can't have those big tails because those are the ones that the cichlids will eat first. So there's always this sort of balancing selection on how wildly can you display without attracting enemies. And in flowers, that's been shown repeatedly over the last 20 years that floral display to pollinators is often checked by third-party enemies that are attracted to the flowers. And for today's talk, the enemies that you need to know about are these little moths, okay? So the genus is Mompha. The family is the Momphidae. They're related to yucca moths, sort of broadly. And they come in 3 flavors ecologically. There are some seed predators that the caterpillars burrow into the ovules, they live in the locules, and then they finish eating all the seeds and they spin a cocoon. So kind of like a yucca moth. And then there are bud gallers, ones where the female moths lay eggs on the bud, the caterpillars go into the bud and then emasculate, actually eat all the sex organs and defecate into the flowers. So there's a huge fitness loss in both of these cases. And then what I'm not showing here is that there's also leaf miners. I think the ancestral condition in the Mamphas are to mine leaves or to spin webs around meristem and then use that as a home. So they're all kind of galling, actually, but they're specialized in the Myrtales, and particularly in Western North America, where the Onagraceae has a radiation with the repeated desertification of the continent, you get these guys showing up again and again in different hosts. Okay, so the Darwin-Nielsen view of coevolution is you have it's binary. You have the moths and their pollinators, and the relationship between tongue length and tube length might co-vary with latitude or altitude, but those are the sources of selective pressure, okay? And these should match each other in nature. And so this is almost like an isometric scaling, right? You scale up if you have a Manduca, you scale down if you have a Hyles in Idaho instead of Arizona, because it's too cold at night for these big guys. And here you're making osamine and linalool at various amounts that are proportional to the surface area of the flower. That would be great if it were true, but it's not. When we did work on this species, Enothera cespitosa, in Jackson Hole, Wyoming, horrible place to work, the Mampha, well, here, Hyles caterpillars, okay, so this white-line sphinx moth is a great pollinator, but there's a price. It lays eggs on the buds of the flowers, and their caterpillars eat flowers, okay? And so there, the flowers are staying open in the morning, they're opening up more broadly, and they're losing linalool and making geraniol instead, and they're pollinated by these huge Anthophora bees. Brilliant, wonderful silver bees with very long tongues. You have to get up at 6 in the morning to see them, but they pollinate every flower. And in Moab, Utah, they're getting hammered by these Mampha guys. And what they're doing is they're slamming shut the flare of the flower, because what we learned is that if the female Mampha can get in and get a drink of nectar, they come running out, they drop into the rosette, and they start laying eggs in the bud primordia. By the time those caterpillars hatch, there's a nice developing bud for them to make a gall out of, okay? But if they can't get that nectar meal, they'll move on to another plant. So there's directional selection here to slam that shut. And they lose linalool as well. So before we even began the study, we knew that there was geographic variation in not only scent, but also flower morphology. And it wasn't just isometrically scaling up and down with pollinator. There's something else going on here. So John Thompson's view of coevolution is about geographic mosaics, where some populations, the predator has a stronger selective effect than the pollinator. And then the flowers respond accordingly in evolutionary time. In other populations, the predator isn't there because it's got a parasite or because there's another host plant that's even better for it. And those plants are released from that selective pressure, and they can more directly match the morphology and the sensory biological needs of the pollinators. And so what Thompson suggested is that there should be a whole mosaic across the populations of these plants where there's mismatched traits with the pollinators and other places where they're matching the pollinators really well, depending on local selective pressures. Okay, so we wrote a grant to the Dimensions of Diversity program and got it on the first go. This is the only time in my life I got a grant on the first try. And this proposal had been submitted as a regular grant for 3 previous years as a pre-proposal, but just slammed. Fair, poor, go away, quit science. This is not transformative, you're going nowhere. And at least I had tenure, right? But for those of you who don't know, the program, which I think has just ceased, is so interesting because it takes the normal amount of funding and cobbles it into a team and extends it from 3 to 5 years. You have 5 PIs, maximum of $2 million. And the criteria are that you study diversification or diversity on 3 different scales, okay? Functional, genomic, and phylogenetic. And so your job as a grant writer is to convince the panel that you've got that and that it matters and that's going to tell a story that any one of those approaches isn't going to tell. And that's what we did. So the way we outlined it was to sort of to model intensive surveying. We're going to choose one focal species in Enothera and really nail it study the whole range. Okay, so we wanted endemic, something that wasn't found over 10 states. But really understand what are the conflicting selective pressures from the enemy and the friend. Are there hot spots and cold spots? Are there mismatches in the traits? If we standardize the way that we collected our data, could we see that, et cetera? And then the second part would be that we would start moving backwards into the lineage and start looking at sibling species or couples of species in different parts of Western North America and ask, do the patterns that we saw here in our focal species actually, are they shared in common across the lineage to which it belongs? And then we wanted to take a really broad look at the end and ask for the whole genus Oenothera, in fact, for the whole family Oenograceae, and for the whole lineage of moths that give rise to Mompha, are they co-speciating? Is this tit-for-tat coevolution? Are they locked in an arms race? Are there other things that we didn't anticipate, like life history strategies or the kind of fruits that they make that might lock out a Mompha moth if it's trying to use fruits as a place to live, et cetera? I mean, Oenothera don't change their flowers that much, but they change everything else. There's no Oenothera trees, but there's lots of diversity in floral form. Okay, so let me focus now on what we learned from our focal study. So we decided to work on this species, Oenothera harringtonii. It's an annual and it's self-incompatible, so we could study its lifetime fitness in a single season. We could grow it, obviously I passed it around. It takes a while to bloom, but they're very, very happy in terracotta pots. They live only here in the Arkansas River drainage. This is their whole world, okay? And there's about 25 or 30 known populations. We hit most of them. So Krista Scogan was the driver of this work. Chris is a conservation geneticist from Ken Holzinger's group at the University of Connecticut. And she got a job. She grew up in Fargo and wanted to get back to the prairie. So she got a job in Chicago Botanic Garden. And we started studying this prairie plant. Here's what it looks like in the field. And so what did we do? We standardized our collection of pollinator data, of herbivore data, of floral scent and nectar and flower measurements across 5 or 6 years, even before we got the grant. We started working in 2008 and 9. And so they are pollinated poorly by large moths because they don't quite fit. They're pollinated extremely well by the white-lined sphinx, which is also laying eggs on them. And then they're visited by these pollen scavenging bees at dawn that do nothing good for the plant. They're one and done. They visit one flower, they carry all the pollen they can carry and go straight to the nest and lay an egg on it. So they are pollen thieves. And across all these different populations, there's all kinds of variation in the relative abundance of bees and moths. But what we found was that there was no variation in their identity. Okay, so also from Colorado Springs and Pueblo in the foothills of the eastern slope of the Rockies, all the way out into the prairie, they're basically getting the same friends, the same enemies, the same ecology, even though there's some variation in the sort of edaphic ecology there. Okay. So our first round, we hit 12 populations, 15 to 30 plants per population. That's a lot of chromatography. And we found something strange, that out in the southeast, the flowers almost never had linalool. Okay, whereas in the northwest and down into Walsenburg, linalool was a major peak. Okay, and so this wasn't subtle. There was a complete drop-off as you leave the foothills and go out onto the prairie. And there could be a lot of explanations for that aren't adaptive, but we don't often see something like this. And we thought, going from a major peak to nothing is interesting. We need to follow up on this. And so we did a couple of unbiased surveys and statistical analyses just to make sure we weren't guilty of circular thinking. We weren't too excited about linalool. There could be all kinds of other things going on here. So we measured everything. What you see here are all the floral volatiles, and everything in gray is a measurement. Flower flare, tube length, corolla diameter, hercogamy, and from all these populations. And then we put them into random forest, which is a machine learning algorithm which tells you what variables are responsible for separating populations out from each other statistically. And so what you look for is a Gini coefficient, and the biggest one by far was our friend R minus linalool. Okay, there's another floral volatile up there, jasmine lactone, and then there's some floral morphometrics. that are giving larger Gini coefficients, but the rest of the volatiles are pretty paltry. Okay, so that's good. How do I know what this is? Here's the chromatography. So Florence is in the foothills, and there's a big linalool peak. There's this mass spectrum again. David's Canyon is out by La Junta on the prairie, and there's no linalool at all. This is presence, absence, okay? And then the rest are kind of typical flower volatiles, osamines, caryophyllene. There's a jasmine lactone that makes these things smell like gardenias, et cetera, phenylacetonitrile. Which linalool is it? We bought a chiral column. Here's the racemic mix from Sigma, R minus, S plus. Here's coriander, which is S plus only. Here's R minus only, which you can buy from Sigma. And here's the Enothera with and without the spike. So it's definitely making, if not completely all R minus, then mostly all. There's a little bump here. Not sure if it's also making a little bit of S plus, but it's hugely dominant R minus. Okay. Is it drift? What's going on in these? We originally studied Enothera harringtonii because that part of Colorado is getting hammered by human change. The cities of Pueblo and Colorado Springs are growing in all directions. There's a lot of light pollution. Hawkmoths can't come to flowers if they're flying around stadium lights and getting eaten by bats. So we thought, oh, there could be some local, drift if these populations aren't getting pollinated by moths. The answer is they are. There's amazing gene flow. FST is tiny across these 25 populations. Anurag Agrawal developed microsatellite primers for Enothera biennis, which is a common weed around here, and they work for Enothera harringtonii. And this result shows that there's huge amounts of gene flow between all these populations. And when we look at the structure analysis and we look at the alleles for these microsats, there's sort of tacit support for maybe 3 conceptual populations, but there's no private alleles, okay? There's no evidence that would make you think, Rob, this is drift, it's a neutral trait, give it up, all right? That's not what's going on here. There's gene flow across this whole region, okay? And this is just another way of doing that. This is QST versus FST, looking at quantitative variation in greenhouse grown heritability type study, comparing it against FST, and only linalool for all the scent traits and floral morphology, only linalool and the jasmine lactone come out looking, significantly higher than what would expect by neutral models. Okay, so it looks like it might be adaptive. The variation in linalool might be adaptive. All right, so let's look at the map. We filled it out. We went back. First year we got the grant. We went back and hit the other populations. So now we've got 19 populations with microsats and floral scent. So the colored pie charts, every time you see yellow, that's the proportion of plants in the population that have linalool, okay? Or the amount of linalool in the floral scent on average. So you see that in the Northwest, they're really putting out a lot of linalool, and that peters out in the Southeast, okay? And so one of the questions was, is there a difference in pollinators? I already showed you a slide showing you that that's not true. And so in fact, when we lump all the linalool producing and non-producing populations together and look at their average visitation rates for hawkmoths and for bees, there's no differences here. Okay? These things are almost uniform across their range. So they have gene flow and they have the same floral visitors and pollinators. So for you to have a different trait in the face of all that also suggests that you've got something adaptive going on. So the next question was, well, what about enemies? Okay, Hyles is visiting the flowers, but they're also turning around and dropping eggs on them. So what if the herbivore pressure from the caterpillars of Hyles might be a problem, as well as Mompha, okay? So then when we look at the caterpillars and eggs, this is eggs of white line sphinx. There's a lot more eggs on the plants when there's no linalool, okay? When there's linalool, there's relatively few eggs. So that's interesting. And here, what we have is Mompha seed predation, so the proportion of fruits that have Mompha in them. And so when linalool is present, there's more of those fruits that are getting hammered, okay? So there's something weird going on here, that linalool might actually help the plant fight against Hyles might be calling the third trophic level. It might be calling in egg predators and first instar larva predators to remove those guys. But linalool may make the plant vulnerable to Montha, okay? So then the question is, you're assuming that they can smell it. You're assuming that they care. You know, go to the wind tunnel and do a bioassay. We can do that with hyles because we have it. Montha is a wild animal. We can't yet raise it. So Jeff Broadhead ran some electroantennograms. And so what these are is here's the response to hexane, which is nothing special, but here's the antenna responding to R plus linalool. Okay, and that's a nice deflection. That's a good sized amount. That's a 1 in 1,000 dilution of linalool and hexane. And here's Montha antennae responding to the same stimulus. Not very much to hexane and very, very nicely to R minus linalool. Here it's responding to a rolled up flower petal in a pasture pipette. So that's the whole blend. So this is just proof of concept that they can smell these things, okay? Doesn't mean behaviorally that they like them, but at least they can smell them.
What are the units on the graph?
What are the units? The vertical units are millivolts and the horizontal are time in seconds, thank you. The red bar is the duration of the stimulus. So we're puffing an odor over an antenna and then stopping the puff, okay? So these are a ½ second each and these are a second each. Thanks. All right. So here, I want to show you a great video that Sol Balbuena collected just a few days ago of how hyles handled flowers. This is a first visit. This moth has never seen this flower before. Okay, and so you can see the amazing contact that it made with the stigma and with the anthers, pulled off a lot of pollen on its legs. We see this in the field all the time, all the time. This was actually done in a wind tunnel. What's the dot? This is a satay spike with a septum on it, and this is the segue into a field experiment that we did where we added linalool to flowers that didn't have them. And so that's what Sol is doing in these experiments. She's flying moths to flowers from the David's Canyon population that doesn't have linalool, and she's adding linalool to half of them and not to the others, and asking if the moths care. Okay. Now here's the other star. Here's Montha. I've shown you still photos of her, but you need to see how she behaves. Okay, look at the antennae. She is drumming her antennae on the surface of the petal. This is at dusk, the same time that hyles visits the flowers. Pollinator and enemy responding to the same cues. Okay. And what she did later on in that sequence is she went all the way down that tube and took a drink, came out and dropped an egg. I wish I had that on film, but we've seen it now 100 times. So what we needed to do was an augmentation experiment. It's hard to knock in traits to a plant that you haven't worked with, genomically, but we certainly could get the emission rates right and add them to the plants out in the field. So this is David's Canyon. It's a shortgrass prairie. It's pretty amazing, open place. We supplemented linalool on flowers every day for about 5 weeks. 70% of the flowers got linalool. We measured herbivory, the number of eggs, the number of caterpillars, the damage. We looked at pollen deposition and removal from flowers. We collected all the fruits to score the presence of Montha caterpillars and their feces and how many, and loss of seeds, et cetera, okay? And this is just to show you that the augmentation worked. All right, so we're adding odor at time 0, and then an hour later we're collecting it. And this is just a slow decay curve over the whole night. And then after sunrise, the emission rates increased a little bit because it got warmer. But basically, these plants continued, the treatments continued to emit linalool through the night. Okay, so it wasn't a 15-minute experiment, which would have been an easy criticism. We nailed not only the first hour when both friend and foe are visiting the flowers, but we kept it going all night. All right, so we're going to use path analysis here to show you what happened. When you add linalool, it increases pollen deposition. Okay, so that's a positive coefficient here, significant coefficient, probably because of more pollinator visitation. We're not sure because we couldn't be there to see all of them, but that's what makes sense. The bees never deposit pollen, so it's probably the hawkmoths. Addition of linalool also significantly increased infestation by Montha. Okay, so these are plants that don't make linalool, and we jacked up the number of Mantha larvae we were finding in the fruits. The other thing it did is it knocked down the number of caterpillars, but not the number of eggs. So adding linalool didn't reduce oviposition on the plant by female hawk moths. What it did is it reduced the survivorship or the presence of the caterpillars once they hatched from those eggs. And we don't know if that means that they left the plant or they got killed, okay, but they weren't there. And then, so here's putting it all together, that linalool has a positive effect on Mantha, which has a negative effect on seed fitness. So this is a classic path analysis, result. The enemy of the friend of your enemy is your friend, et cetera. And it suggests that what we're seeing in the southeast part of the range where we did this experiment is that linalool is a liability, that if flowers were to make it there, they would incur fitness loss by more seed predation, okay? So we went back to the corridor where the Mantha infestation was highest. And that's this road down here, Interstate 25, down to Albuquerque, okay? And these populations down here had the highest Mantha infestation. So we went back the next year, 2017, to these 4 populations or 5 populations, and we did a correlative study. This is a phenotypic selection study where you regress fruit set and seed fitness against everything you can measure: flower size, flower number, linalool emission rates, et cetera, okay? And here's what we found. There are 5 populations all over here in this sort of death alley from Mantha. All these other traits: corolla diameter, flare, tube length, hercogamy, nectar volume, sucrose, the major 4 compounds. None of these departed from null expectations in any of the 5 populations except linalool. And it happened only in Raus, which is right there. And what was funny about Raus, it was that it was an allele effect. It was a small population with small plants. So what we think is happening is that a large, robust plant with 80 flowers per year can tolerate damage from Mantha moths because it's still going to make 50 or 60 fruits. But a small plant in a bad year with no second chance because it's an annual and no ability to self-pollinate, that's when the screws get tightened. So that's what we think after 5 years. That's what we think we're seeing. It's really hard to do this. And coefficients are not very satisfying. But this is all the assays together lead us to believe that this is what's happening. Okay. And so in sum, plant size and whatever abiotic factors influence plant size is probably setting the context for whether this hurts that, okay? So let me finish by asking, if we scale this up outside of Colorado, outside of this one species that we obsessed with for 5 years, do we see anything else that's useful? Do we see these patterns propagating across the American West? Do we see, in John Thompson's parlance, a geographic mosaic of coevolutionary processes? So here's the family Onagraceae. I'm sorry for the lack of confidence values, but this is a cartoon, just to show you that the ancestral condition for the whole group is almost certainly bee pollination, with 1 or 2 transitions to hummingbird pollination. These are not indicative of a species richness. Fuchsia is a huge genus. Epilobium is a huge genus. Just focusing on Enothera here. So Enothera itself probably was ancestrally bee pollinated. We have strong support for a lot of these transitions here. And it looks like there was 1 major transition to hawkmoth pollination at the root of many of these Enothera lineages. And then they translate to self-pollination or noctuid moth pollination or back to bees. So we wanted to capture some of this diversity phyletically. These are some of the species that we worked with, including Clarkia, the plant I worked with as a grad student, with 1 species out of 45 that's hawkmoth pollinated. Okay, so there's several different clades represented here in our plan. So what we wanted to do was hit 15 different species, strategically chosen to represent, couplets, closely related species whose biology we suspected was going to be very different. And here's their relationships phylogenetically. And what you see here is that for each color bar, that's a species. And each one of them had 3 to 5 populations that we went out and sampled over 5 years. And so they're all color-coded across the American West. Now again, not a bad place to work. Pretty amazing. There's no normal years, right? Every year is different for rainfall and for the early onset of blooming, or a great monsoon and everything blooms again, you know, in the Chihuahuan Desert in August. The first year of the project, we had a September bloom. You just got to run and do it. But what we wanted to do was capture some of that population level variation that we had seen in Enothera harringtonii out here. Try to get sort of smaller versions of that variation in as many other species as we could. And so our approach was, again, to bring out large teams and standardize the data collection. Floral scent, morphology, nectar, pollinator visitation, herbivory, Hyles eggs and larvae, Mampha in the buds and in the fruits. We worked in 58 populations, many, many states. This is a lot of work. Most of the budget of the project. And yet, the players are so similar across the whole family. The same enemies, the same friends. And so here's a quick look. The linalool polymorphism extended well beyond Enothera harringtonii, differs among sibling species and among populations with different species of Enothera. So there's a couple of independent origins of S plus linalool. We knew about Clarkia breweri because that's what I'd studied as a grad student. But there's another one in the section Callilophus that accompanied a switch from Hawkmoth ancestry to bee pollination in West Texas, okay? Then there's a couple of examples here where an ancestry is likely a linalool producer, and there's been an inferred loss of R minus linalool, okay? Here, this is a switch to bee pollination. This one here is still being pollinated by hawkmoths, like some of the populations of Enothera harringtonii. This one here, Enothera brachycarpa, has the largest flowers in the genus. It makes no scent, and it makes no sense. It is almost entirely vegetatively propagating at this point, and it's being destroyed by Mampha. So we don't know how they're finding the flowers, but they've almost given up on pollinator display. And so the question we asked is, if you take all of this data, can we ask to what extent pollinators versus Mampha are structuring some of the data, the variation in the data sets? So this is we're going to do a phylogenetically controlled multiple regression, okay? And taking into account phylogenetic distance, geographic distance, pollinator distance, and which species of Mampha. And the coefficients we got for pollinators were both for morphology and scent. And for Mampha, interestingly enough, even though we know that flower trumpet flare matters to Mampha, at this phylogenetic scale, they were explaining more variation in scent and not so much in morphology. So we had the right to look at it now. This is basically a principal components analysis of flower morphology across all of our taxa. And what you see here are the bee pollinated ones are clustering up here in a morpho space. The Hyles, small, medium-sized hawkmoth pollinated ones are clustering over here. And then the ones with really long tongues and tubes are clustering down here. Okay, so this is pollinators are very nicely explaining variation in morphospace, although each of these dots is a different population, and certainly in many cases, there's considerable variation between populations of the same species, okay? For scent, these are all hawkmoth pollinated species, and they're all over the board, okay? So some of them are making R-linalool, but that's not a strong predictor. There's nitrogenous compounds down here, there's S-linalool up there. And look at also the spread in morphospace or in chemical space of these populations of the same species. These guys are close, these guys are not, okay? Whereas the bee pollinated ones are off in the periphery. They are really leaving the chemical space. So between them, we think we have a lot of structure here. Here's what Mampha does. All right, so here's the same phylogenetic relationships. The pie charts, if they're blue, mean that that species, at least in some populations, has R minus linalool, which we've been talking about all the time. If they're yellow, they've switched to S plus linalool. And we don't know what that means. It attracts moths, but we don't know if it attracts Mampha. Almost done. And then if they're gray, they've lost linalool entirely. And so here, the black bars show how many populations in species X has infestation from Mampha. Some species are free of Mampha. others are not. And then we're tracking the different kinds of Mamphas, the flower bud galling ones and the seed capsule galling ones. So the good news is there's an awful lot of stuff going on here. The bad news is there's not a clear direction of one Mampha species driving flowers to be silent, removing scent entirely. And in fact, there's other species that are shifting to diurnal bee pollination and are still getting hammered by Mampha, okay? You would imagine that maybe those flowers would be under selection to close at night when Mampha are active, okay? So to wrap up, what have we learned? Linalool emission is certainly a labile trait within population species and sections of Oenothera, and I've shown you that it's found across the world in all kinds of other angiosperm families. Linalool appears to be selected against by seed predator Mampha moths without sacrificing Hylie's pollination services, which leads to the question of why is linalool needed at all in Oenothera harringtonii and similar species? It might be a phylogenetic artifact. It's a shared derived trait in that group of Oenothera. Escape from Mampha might involve the loss of linalool, the switch to a different enantiomer of linalool, switch to bee pollination, or switch to self-pollination. And herbivores, in sum, clearly play important roles and complex roles as selective agents on floral scent variation. So I want to thank the big team that we worked with in the field over 5 years, Amherst College, Chicago Botanic Garden, and Warren Wagner from the Smithsonian. And certainly Jeff Broadhead, Sol Balbuena, and Deidre Jacobson for helping us to study some of these interactions here in Cornell during the year. And our funding sources and cooperators. And thanks very much. Look forward to questions.
You had some species of genera there with no Mampha.
Mm-hmm.
Is there any evidence of chemicals which are repelling as distinct from attracting?
Right. So this is another prediction of the geographic mosaic, that not all odors are attractive, and there should be strong selective pressure either to hide or to push away. We have to do the experiments. It's interesting that the second compound that showed up in the QST FST analysis was jasmine lactone. It's not a common compound in flowers, except in jasmine. But lactones have a repellent quality. In insect control, lactones can often be repellent. So that's a compound that we could actually add to flowers and see if we reduce Mampha attack. Several of the ones that had no Mampha attacking them actually produce lactones. It's a good question.
You transplant from one region to another, how long would it take for the plants to switch their linalool?
Yeah, that's a good question. I think that they have enough standing variation in each population that the selection could act pretty quickly. We were really sensitive to the problem of not going to the populations that make linalool and turning it off, right? I mean, that would be the Ian Baldwin way to do it, is to use gene silencing or to breed, the few individuals in those populations that don't make linalool. Of course, there could be all kinds of other things that are coming along with them, so you really do want a rill. It would take a decade to make rills with these guys. But a reciprocal transplant would be interesting. I mean, ethically, you would want to remove the pollen, I would guess. But you certainly could look at Mampha attack on plants from the prairie that were grown in Colorado Springs versus plants from the Colorado Springs that were being grown out in the prairie. To water them every day would not work. It is a parched place. Sol and I went to work there this spring, and it was an unbelievable drought. There were no bees, just a few plants blooming. It is the fate of the West at the moment, and prairie Colorado is becoming the Chihuahuan Desert. So, but we are aware of that, and we wanted to do reciprocal transplants. That would have been a really nice experiment to do. Yeah.
That was a nice talk. What about so in the graphs that you showed the amount of linalool versus other compounds, but that doesn't indicate the actual quantity of linalool. Is that correct? So is there
yeah, pie charts are crude. That's a great question. What we in the publication that we're about to submit, we quantify, so what we do is we always have an internal standard in our chromatography just to make sure that we haven't injected the wrong amount. But then we run external standard curves for all the major volatiles, and we quantitate them independently. Because in mass spectrometry, you have really different response factors to the 70 electron volts, ionizing radiation. So energy rather. So yeah, we quantitate them all, and we try to eschew percent of total. We try to actually present nanograms or micrograms per flower per hour or per gram of flower per hour.
Was there a lot of variation in the actual amount? I'm just thinking about linalool potentially having a different effect in different concentrations. Yeah,
that's a great question. The dose often makes the toxin or the attractant in chemical ecology. What we found is that there are plasticity issues. When we grow the plants from Florence, which make huge peaks, in our greenhouse with no UV, okay, those peaks fall fourfold in peak area, okay? So we were conservative. The amount of linalool that we added in the field was the greenhouse amount and not the field amount. We were worried if we added too much linalool, we'd have some artifact of toxicity. So we actually chose, we lucked into actually an amount that we could add in the evening that constituted a conservative test. Yeah. No, but that's an important question. In chemical ecology, you have to be very careful, which is why expressing a transgene in a plant, with an appropriate amount of regulation, could get you the phenotype you want, if you're lucky.
Very interesting story. I was thinking that it's not only synthesized in the flower or any other part of the plant.
Great question. We did a transcriptomic study, which I didn't show here, looking at expression in stamens, stigma, petals, buds, leaves, etc. And we have all kinds of candidates for an R- linalool synthase from this plant. The leaves don't emit very much. There's some screaming candidates in leaves, but we don't get a lot of headspace coming out of them. Other plants do. We're working with jewelweed right now, which makes linalool in its leaves and not so much in the flowers. But in this species, the petals are the source of linalool. The nectar tubes are 9 centimeters long, and there's methyl benzoate in the nectar, but there's no linalool in the nectar. They're really separate functional parts of the flower because of that distance of the hypanthium. So it's a good question.
You mentioned that linalool production is highly influenced by environment, as shown by greenhouse plants producing much lower amounts. And you show this map of linalool variation in production. Have you considered factors such as environment, UV, radiation, and kind of like levels?
Yes, thank you. Excellent. We did. We worried. So these are the linalool producers. These are the linalool non-producers. This is annual average high temperatures. Okay, across. I guess we have 3 or 4 populations of each. We didn't get weather for all of them because some of them are literally in the middle of nowhere. But precipitation, there's no pattern. Temperature, there's no pattern. Winter precipitation, there's no pattern. We also looked at a principal components analysis of soil. Okay, they grow in silt, really fine grains. And we looked at all kinds of macronutrients and micronutrients and particle size. No clear explanation there.
Is there any change in, I know it's the prairie, so it's pretty flat, but is there any change in like altitude?
Yes, absolutely. Florence and Pueblo, Colorado are coming up off the prairie. They're probably like, Colorado Springs is 7,000 feet up, right? It's higher than Denver. Whereas out in the prairie, it's like 800 or 900 feet above sea level. So there's a considerable drop in elevation.
So yeah, I guess my thought was that could sort of explain it, because a lot of time greenhouses have lower production of secondary metabolites because they're receiving different UV radiation because of the coatings on greenhouses.
Yeah.
So, if you have difference in elevation, you have difference in atmosphere, you're getting different wavelengths.
Yep, lots of UV out there. That's a very good point. What's fascinating is that all the other scent compounds don't change. Linalool seems to be the one that responds to UV. But it's an excellent point, thank you. Yeah. So I should write a grant for UV lamps. Yeah. All right. Thanks.
This has been a production of Cornell University, on the web at cornell.edu.